Okay, so today I will be talking about the cell cycle and mitosis, and this is part of topic 1.6 of the IB biology syllabus. So first of all, mitosis is the division of the nucleus into two genetically identical daughter nuclei. Now in GCSE we usually say daughter cells, but it's important that we recognize that mitosis is in fact the division of the nucleus into two genetically identical daughter nuclei. The importance of mitosis. Many unicellular organisms reproduce this way. It is a form of asexual reproduction, and they use mitotic cell division for numerous reasons. For example, for embryonic development, for growth, the repair of damaged tissue or cells, and for asexual reproduction. The cell cycle. So there are three phases of the cell cycle. We have interphase, which is the period where the cell is not dividing, when the cells increase in mass and size. And then we have mitosis, which is an active division, and cytokinesis, which is the division of the cytoplasm. So we can see in this micrograph two cells, one containing chromatin and one containing chromosomes. So we need to understand the difference. Chromatin is unwound DNA, and it is found throughout interphase. Chromosomes, on the other hand, is coiled up DNA, which is found during mitosis. And we can see the difference in both cells. So we can see that here there is a diagram of a chromosome which is made of two identical DNA molecules which are known as chromatids, and these are connected by the centromere. Interphase. So during the cell cycle, 90% of it is taken up by interphase. So when looking at a micrograph of cells which are undergoing cell division, most of these cells will be in interphase. So the cells carry out the normal cellular activities, for example, synthesis of proteins, enzymes, etc. DNA replicates and the chromosomes are copied. So there are three phases of interphase. First we have G1, in which the cellular contents duplicate except the chromosomes, and the cell grows. Then we have the S phase, which involves DNA replication. And the third phase is G2, when the cell double checks the duplicated chromosomes for errors. So S phase is DNA replication. And we'll look at a mitotic chromosome. So as I said before, it is made of two sister chromatids with narrow centromeres, and they contain identical copies of the original DNA. So during the S phase, the chromosomes are duplicated, and another chromosome is formed, but this one has two chromatids, and it forms an X shape, and both chromatids have identical copies. They're identical in terms of their DNA. So as you can see, during the S phase, one chromosome is copied into another chromosome which has two chromatids. So each strand is a chromatid, And this is one chromosome, this purple one, and this other purple one is also a chromosome, but this one has two chromatids with two copies of the same DNA, if that makes sense. Okay, so here we have an animal cell and a plant cell which are undergoing interphase. So in mitosis, we have 4 phases. First, we have prophase, metaphase, anaphase, then telophase. An easy way to remember this is to just remember PMAT, the word PMAT, prophase, metaphase, anaphase, telophase. So first we have prophase. The chromosomes become shorter and fatter by supercoiling. The nucleolus breaks down. The microtubules grow from structures called microtubule organizing centers, so centrioles, to form a spindle-shaped array that links to the poles of the cell, and the nuclear membrane breaks down. So in this phase, the spindle fibers are formed. So we're able to recognize this phase because we can see that the chromosomes have started to become a lot shorter and fatter, and this is seen in the micrographs of the animal and plant cells here. The second phase is metaphase. So the microtubules continue to grow and they attach to the centromeres on each chromosome. 2 attachment points on the opposite side of a centromere allow the chromatids of the chromosome to attach to the microtubules from different poles. So when the chromosomes line up at the equator of the cell, the spindle fibers attach to the centromeres, and each sister chromatid is attached to a spindle fiber from the opposite poles of the cell. The microtubules are all put under tension to test whether the attachment is correct, and if so, the chromosomes remain on the equator of the cell. So we're able to recognize that the cell is in metaphase because we can see the chromosomes lining up in the center of the cell. And the nuclear envelope has already broken down, as seen in these micrographs. Third phase is anaphase. So each centromere divides, allowing the pairs of sister chromatids to separate. The spindle fibers contract and the sister chromatids separate. The spindle microtubules pull them rapidly towards the poles of the cell. So here we can see an animal cell and a plant cell which are currently in anaphase, and we can see this because spindle fibers are pulling the chromosomes to opposite poles of the cell. Lastly, we have telophase. The chromatids have reached the poles and are now called chromosomes. The spindle fibers break down at each pole. The chromosomes are pulled into tight groups, and a nuclear membrane reforms around them. The chromosomes uncoil and the nucleolus is formed, and the centrioles are also reformed. So here we can see quite clearly that the cells are in telophase because two genetically identical nuclei has been formed, and this is seen as the chromosomes have uncoiled and they are now in a nuclear envelope. So now that we've looked at mitosis, we'll look at the third phase of cell division, which is cytokinesis. Cytokinesis is the division of the cytoplasm, and it usually occurs simultaneously with telophase, which is the last phase of mitosis. Cytokinesis is different in animal and plant cells. In animal cells, a cleavage furrow is formed. A ring of contractile fibers tightens around the center of the cell, allowing the cytoplasm of the animal cell to divide. And in plant cells, a cell plate is formed. The cellulose cell wall builds up from inside of the cell outwards. So here we can see the division of the cytoplasm in both animal cells and plant cells. So hopefully now you should know the different phases of mitosis: prophase, metaphase, anaphase, telophase, PMAT. Okay, so now try to test yourself and try to state which phase of mitosis each cell in the micrograph from A to H is being presented. Pause the video and try this yourself. Okay, so here are the answers. And that's it. Thank you very much for watching, and please remember to subscribe to our channel for more videos like this.